Thank you to the organizers, particularly Arthur, for this wonderful introduction to your country, which is beautiful and historical, and I really appreciate the perfectly organized conference and opportunity to give this talk. I'm part of the myeloma group at Sinai, and my focus is more on translational aspects of this disease. I'm hoping to give you a picture of what we're doing, and I'll try to make it clinically interesting with some patient vignettes and take-home points that may be applicable to your practice as well. So, as you know, in myeloma, we currently classify the disease based on certain biochemical features and cytogenetics. But these classification systems are not perfect. We have patients that have low-risk disease that do badly and vice versa. And we have not had the benefit of having a comprehensive molecular profiling done like other solid tumors had with the Cancer Genome Atlas. Because of this, a few years ago, there was a huge effort in the US and now internationally to collect over 1,000 patients and follow them sequentially, collecting their genomic information and clinical information simultaneously over a period of time. And this effort has now developed a data set of next generation sequencing data with whole exome and RNA-seq data that is really impressive, because it's actually bigger than the largest cohort in the TCGA, which is for breast cancer. So this is over 1,100 patients now, and we are following patients from newly diagnosed setting onto relapse and beyond. So what do we do with all this data? And in fact, the data is so large, we actually have to house it on our special servers or web-based interfaces to even analyze it. The approach we've taken to analyze it was in part developed at Mount Sinai by a mathematician who used a similar analogy to what companies like Facebook and Twitter work, like social networks, but he developed a system for looking at gene co-expression networks. And in this, the explanation that I can give you is that the expression of genes that are related is in function are generally together. And based on this, when we have a large data set of RNA-seq data, we can actually start creating what is similar to a Google map of disease by taking a city and dividing it into neighborhoods, and then annotating the neighborhoods for different useful aspects. For example, in a typical map, you would have different neighborhoods here. These are gene neighborhoods shown in different colors that we then annotate with useful clinical traits such as stage of disease, what treatments may be important, what the pathways are related to the genes in that area. So in that way, we can design neighborhoods that have useful information. Are these good neighborhoods or bad neighborhoods? And how does this help segment the disease better and treat patients better? So we undertook this exercise with the first about 450 patients from this study, and we found that in myeloma, there were 37 distinct neighborhoods of genes. And when we annotated these neighborhoods, we were able to actually correlate different neighborhoods with different genetic characteristics and clinical characteristics. And it was very interesting because some of the known biological features readily came about, and we found new and interesting things which I'll share with you. We published this earlier last year, and we are now refining this work as the relapse data comes about. But I'm going to highlight a few vignettes from the network that we've developed. I'm going to talk of one neighborhood which is called M9, where actually the expression of the genes in M9 correlated with a lot of the bad things that we know about myeloma, correlated with advanced stage of disease, bad cytogenetics, high mutation burden, p53 mutations, so on and so forth. In fact, as you can imagine, high expression of M9 resulted in a bad prognosis. To make this a more readily usable test, we actually narrowed down the gene signature to a panel of just 4 genes and validated in 2 independent cohorts of over 1,000 patients that the expression of these 4 genes actually can reliably separate patients that have differential outcomes. Another application, and this was quite surprising, I think there are some lab people in the audience that may be able to appreciate this. So typically we segment our myeloma patients into high risk on the basis of cytogenetics and FISH. And over here we compared FISH-based identification of patients that have MMSET translocation to whole genome and RNA-seq-based identification of these patients. As you can see, this in red is the expression of genes that are high in the patients that have MMSET translocation by whole genome sequencing. And you can see in green that most of these patients have also been labeled by FISH. However, there are several white spots in here where patients were mislabeled as not having the translocation, and what was very concerning is there were patients without MMSET by whole genome sequencing that were labeled 414 translocated by FISH. So we are trying to find signatures that can be used to better identify these high-risk patients and actually find new targets for them. Similar to social networks, gene co-expression networks can identify hierarchies of genes. And to our surprise, we expected MMSET to have the highest point in the hierarchy, that is the most connection within its module of genes. And instead we found 2 other genes, CDC42 and CLEC11A, as having more connections or higher in the gene hierarchy. Now, these have not previously been described to have a role in myeloma, and we wondered what they were doing here in 414 myeloma. To interrogate the function, we actually knocked out these genes in vitro in 414 cell lines and found that they not only caused significant cell kill, but they also seem to regulate a very important gene downstream, MMSET and NF-kappa B. So we found new regulators and established hierarchies of genes that were novel just by interrogating the structure of the gene modules in this network. Lastly, we were able to actually segment the disease, and it recapitulated a lot of the classifications that have emerged over the years, the TC classification, the GP-based classification from Arkansas, for example. But we also found several new classes of disease, patients that had MYC mutations, for example, patients that had high activation of cytokines that may be more amenable for immunotherapy, for example. And these may be groups that are more important down the line. We are now following these patients as they progress and relapse. And what's interesting is if we use the whole exome sequencing data and model the clonality of these patients, we are finding that on average, most of these patients have 3 to 4 clones that are completely genetically different and behave differently with chemotherapy. And while we are treating the patients and continuing the treatment, there are some clones that seem to respond and some clones that are completely blowing through treatment. So really identifying the genes and understanding what is the clonal composition of a patient is going to be very important down the line to design treatments that are more specific against the drug-resistant clones. I'm going to skip my conclusion slide to move to the second part of the talk is about a project that I took on when I came to Sinai. We have a large group of patients that are referred for complex myeloma, actually patients that have relapsed multiple times. And this lady, many of you all may know her. She's one of the founders of the Myeloma Foundation in the US, and she's herself a myeloma survivor who got an allo transplant from her sister and is still around and doing great work in fundraising and developing projects such as Compass. So her statement here is, next-gen treatment shouldn't take a generation to get there was very important, and struck me that we need to find these biologically driven treatments to patients in a time-sensitive manner. To do this, we actually set up a pilot study to use next-gen sequencing of DNA and RNA to interrogate several large databases that have been generated that look outside of the typical myeloma toolbox for drugs that are either FDA approved in other cancers or in non-cancer indications that can be repurposed for myeloma. And I'll present to you some examples. So typically, the patients that went into this pilot study, this is a snapshot, were extremely refractory. You can see majority of them were more than 5 lines of treatment, some of them even more than 10 lines. They had all received PIs, IMiDs, autotransplant, daratumumab, and they had bad prognosis by cytogenetics and stage. So what did we do for them? We took their bone marrows and isolated their tumor cells, separated them for DNA and RNA sequencing, and then we've developed actually an algorithm that comes out with a physician-friendly report to help clinicians in clinic think about drugs that are outside of myeloma toolbox while they are seeing the patients when they are relapsing. And to do this, we actually have a special server, and we have a pharmacist in clinic that helps us get these drugs for these patients. We've described the methodology and initial results in a small paper that came out earlier this month. And this is what the reporting portal and reports look like. So we can pick a patient, and typically we have longitudinally biopsied and sequenced these patients, so we can pick a particular report. And the report looks like this. So for a particular patient, we have that these are the mutations that the patient has, these are the treatments that the patient may be sensitive based on mutations. Also, we look at copy number alterations and what treatments patients may be particularly thought of. The thing that we are doing that goes a little bit beyond what targeting sequencing platforms are is we are looking at RNA-seq and databases that use RNA-seq to find drugs. And this is interesting and novel, and we've been able to actually use this information in a surprising way. So we use the RNA-seq information to not only calculate known features like the GEP70 score, which is an estimate of how aggressive a patient's myeloma is, but we also look at biomarkers. Earlier, for example, David had mentioned BCL2, and MCL1 is one of the biomarkers for BCL2 inhibitors, so we can actually read these out. We can also read out BCMA. We've found actionable mutations and copy number alterations that are described in our paper that were interrogated using a standard database, which is called CIVIC. I'll give you a couple of patient anecdotes to see how we are evolving this system to help. And one of the classic and common mutations that we see in myeloma are mutations in the MAP kinase pathway, NRAS and KRAS. We had a patient that was failing most FDA-approved treatments and had a particular mutation in NRAS that substitutes glutamine for arginine at codon 61. This is a known activating mutation that has been described in melanoma, and there is a drug that is FDA approved for the treatment. It's an oral kinase inhibitor called trametinib. So we gave the patient trametinib, his M-spike responded, he did well for several months thereafter, and then went on to a clinical trial after this. He was actually interviewed, and his interview's on YouTube if anybody is interested in looking it up. He's a physician himself and still around in New Jersey. Our computational pipeline also looks at RNA, and I'm going to give you a couple of examples of how we use this to help patients get treated. The RNA-based identification comes up with a number of pathways that are activated in an individual patient. PI3 pathway, HDAC inhibitors, MAP kinase, and others have actionable drugs. This is one patient that was actually, after multiple relapses, going to hospice, and we sequenced her tumor as a last-ditch attempt. and found in multiple databases sensitivity to an HDAC inhibitor. Panabinostat is approved in the US, but we don't really use it much in our practice. When we actually treated this lady with panabinostat, she had a dramatic response in terms of her tumor, and in fact, instead of going to hospice, she went for a vacation to Hawaii. So when she came back, she lives in Connecticut, and Connecticut is a very woody area. So while walking through the woods, she contracted a tick bite and developed Lyme disease. And we had to actually stop her treatment for a little bit, and you can see her tumor starting to come back up. Upon finishing the antibiotics for Lyme disease, we re-challenged her with the HDAC inhibitor, and indeed, she responded again, went on to respond for several months. We are now evolving the system to give multiple drug recommendations. I'll give you an example of that. This is a patient that comes to my clinic every other week. He's an artist who still paints every day, and he was essentially going to hospice in January this year because he has, along with myeloma, cirrhosis of the liver, which precludes him from going to a lot of clinical trials. So we actually sequenced his tumor, and based on the sequencing, he had sensitivity to not only trametinib, which is also known as Mekinist, I've described that before, but also a drug that's used in breast cancer called Ibrance. It's a CDK4/6 inhibitor. So we gave this patient a cocktail of drugs based on the in silico prediction, and his tumor dramatically responded and continues to respond even now. He's still around, he's continuing to paint, he still comes to my clinic every other week. It's now 8 months since he was supposed to be in hospice, so there was some tangible benefit to this, and we are continuing to evolve this. The way we think that this is going to be useful is, number one, I think most current precision medicine algorithms in the US are looking at targeted DNA sequencing alone. This has to change. I think our study is initial proof that RNA-based sequencing is also useful. For example, if we look at a series of patients that were treated using this methodology, this is the reduction in tumor that was seen. The patients in orange are the ones treated based on an RNA-based methodology. The ones in blue were treated based on a DNA-based methodology, and you can see the ones in orange far outnumber the ones in blue. This treatment is not perfect. The current duration of remission is short. It's to the tune of a few months. I think as the system evolves to make more complex decisions, such as combination treatments, we also can use it to send patients in a more biologically rational manner for clinical trials. We can also use this to actually look at patients with deeper sequencing methodologies like single-cell sequencing. I'll give you an example in just a minute. We did learn that we need even faster turnaround times. The current methodology using the HiSeq machines takes about 4 weeks in our core facilities. We are now purchasing a NovoSeq machine, which is much faster. We're getting a turnaround time of 2 weeks and hoping that's going to help more patients. And to get to more clone-specific predictions, we are using single-cell sequencing. The initial methodology we used for this is one patient whose tumor was sequenced, and this is a snapshot of about 5,000 single cells from the patient's CD138 compartment. You can see each different color actually represents a transcriptionally different clone of disease, which has different pathways activated, and really to treat a patient that has so much intra-patient heterogeneity really needs a targeted approach that combines many different agents. Besides the tumor, we are also using single-cell methodologies to look at the microenvironment. Besides single-cell sequencing, CYTOF is a methodology that uses heavy metal barcoding instead of fluorescent labeled antibodies to look at multiple analytes in a single patient. We are combining actually single cell and CYTOF in a new technology called ProCodes, which is going to come out in a couple of months, to look at tumor microenvironment along with the CD138 compartment at the same time. The last part of my talk, I'm going to talk about how we are repurposing the data that we're getting from sequencing to understand how to use immune approaches better. And my lab is right next to a cancer vaccine lab that actually develops different platforms. I'll describe the general methodology to you and how we are using this. So when patients have mutations in the DNA, a subset of these mutations are immunogenic. These are expressed in the context of the patient's MHC system and can be recognized by the immune repertoire to actually therapeutic effect. I'm going to show you an example of how we use this, not just to make vaccines, but also apply different therapeutics. So this is the pipeline, how we develop the mutanome, and we take the mutations that the patient has, read out the HLA from the genomic sequencing data, and subset the mutations of what mutations may be expressed at high levels by the patient's HLA system and are capable of being recognized. In the vaccine approach, we can develop overlapping peptides that are immunogenic and correspond to these mutations, but this is what a typical patient's sequencing report looks like for the mutanome. So that we get the HLA information, we get the peptide that is immunogenic caused by a mutation, and this is the IC50 or the binding potential to T cells that is predicted from the in silico prediction. This is the mutation location within the gene. And like this, we get a readout typically of 40 to 50 different immunogenic mutations that are there. We've looked at about 200 patients, 100 with newly diagnosed myeloma, 100 with relapsed myeloma, and majority of the patient have more than 50, some have even several hundred Neo-antigens that are immunogenic. So really there is a potential for harnessing these Neo-antigens. I'll give you an example of how we are doing this. This is a patient that was similar to a case that was presented earlier, primary refractory myeloma, very aggressive disease. She was referred to us from colleagues in Connecticut after progressing through carfilzomib, pomalidomide, and dexamethasone. In fact, she received even salvage chemotherapy with DSEP, received targeted kinase inhibitors. It didn't really last. And the patient was blowing through. We decided to use, as a last stage salvage for her, a novel approach that was being developed in our lab, which is called immunotransplant. This is a concept that came out of Ron Levy's lab at Stanford, actually, where we used the lymphopenic period following the transplant to educate the immune system against the patient's neoantigens. And what we did was we gave the patient a couple of doses of anti-CTLA4, anti-PD-1 combined therapy, gave her reduced dose melphalan BCNU myeloablation or lymphodepletion, if I can call it. And then she went on to get ipi-nivo again, and then nivo for a couple of months combined with Revlimid. Patient went into stringent complete remission. And we can see here, this is the T cell sequencing of her peripheral T cells. You can see an increase in the T cell clones that are emerging following this immunotransplant maneuver. But this is not proof that the T cells that we are finding are actually reacting against antigens. So we went to the next step and created an experiment where we took each and every neoantigen that she had and interrogated her own T cells before and after the immunotransplant maneuver to see if the T cells recognized the neoantigens. So she had 40 neoantigens, and we started looking at each of them systematically and found that there was one particular neoantigen in a gene called PRKDC, which was immunogenic in her case. If you look at the readout here is actually secretion of gamma interferon, TNF-alpha, and IL-2, classic cytokines that are released by activated T cells. We find that the post post-treatment T cells are strongly reacting to this mutant neoantigen. As a control, we actually use the same sequence without the mutation, and we find that there is no response. So this is a very specific neoantigen-specific T cell response that was elicited in this patient. Moreover, we then took these T cells and fished them out of her blood using tetramers that was specific for her HLA and sequenced them, and we find that they are not only increased in clonality, but they actually kill the patient's autologous HLA-matched cells. So we did a killing assay, and these are the negative and positive controls in the killing assay. And the killing was specific for the mutated T cells and not for the wild type cells. So we are hoping this sort of approach, maybe not in combination with IMiDs, but maybe in combination with vaccines, could be useful early on in the MGUS phase, or even later on. As we are seeing from colleagues at UPenn who are using checkpoint inhibitors post-CAR T. Dr. Raj is also using it post-CAR T in myeloma. I think this has an opening. Here we are now finally able to identify the mutation-derived neoantigens using next-gen sequencing, which is very exciting. So with that, I'll conclude and thank my entire team. We are trying to build a learning system where we take the patients and learn from them with different platforms to develop personalized medicine for myeloma. It's clearly a group effort, and we have to thank all the physicians I'm working with, especially Sundar, who recruited me to Sinai. There's a big computational team, members of my lab who took part in this, and the patients who gave their samples for this. So with that, I'll stop and take questions. Thank you.